Hi everyone, welcome to the Bionano Genomics Genome Finishing webinar series. All the webinars in the series are pre-recorded. After the presentation in each webinar, there is a Q&A session to address most popular questions. My name is Lucia Muraro. I'm Field Application Specialist at Bionano, and I will be the host for today's webinar. Today, we will hear the story from Dr. Lucy Poveda. Her presentation title is Exploring the Use of Bionano Optical Mapping for Genome Assembly. Lucy is from the ETH and University of Zurich in Switzerland. She's a senior scientist at the Functional Genomics Center of Zurich, FGCZ, which is a state-of-art technology, research and training facility that concentrates on high-throughput omic technology. Lucy has an extensive experience in the field of genomics technology and has been working and supporting numerous projects in the scientific community of not only Zurich, but internationally as well. 3 years ago, she implemented the Bionano optical mapping at the FGCH Institute, which she has used for a wide range of genome assemblies, including vertebrates and plants. Without further ado, Lucy, the stage is yours now. Thank you.
Thank you. I would like to really thank you for inviting me to this webinar series to share our experiences on the use of optical mapping for genome assemblies. So the FGZ actually focuses its expertise on high-throughput omics technologies, so in a wide variety of genomics, transcriptomics, proteomics, and metabolomics using next-generation sequencing and mass spectrometry. So as you mentioned, in the recent years, we implemented alongside our long and short read sequencing Bionano optical mapping as an orthogonal complementary technology. So first we started using the iris, and then we went on to use the sapphire. And from this on, we supported a wide variety of projects. So since its implementation, we worked in a wide variety of genomes, ranging from different complexities and also sizes. So here you can see a little bit some of the organisms we worked up to 2019, and we actually have about 6 to 8 further organisms which are in the pipeline. I would actually like to draw this analogy between assembling a genome to actually putting together a jigsaw puzzle. So you've got these millions of different pieces. Some of them are smaller, some of them are bigger. And the challenge is just to assemble them together, where you might have, so you do not really have, this box or picture to refer to. You have perhaps an estimation on the size, for instance, through flow cytometry. And it is, or you might have, for instance, a close relative as well. But actually, there's a very important fact, which is the actual underlying genome, which can actually bring a lot of complexity, such as heterozygosity levels, repetitive elements, and different also ploidy levels. And so for the outline of this talk, I chose 4 different projects with very diverse underlying genomes and that used different sequence assemblies. And I would like to show you how optical mapping enabled these genome assemblies. Also, after that, I would draw some remarks or out of our experiences, really so some thoughts on the different, on how we could use this technology, and then some conclusions. So the first project I'll discuss followed mainly a short read assembly. And this was done for the song sparrow, which is Melospiza melodia. So this project was done together with Professor Lucas Keller and Glauco Kamenisch from the Department of Evolutionary Biology and Environmental Studies at the University of Zurich. This bird is common in North America, and it's a model species for acoustic learning and memory, and it has a huge song repertoire. So its karyotype is about 38 to 40 chromosomes, and we have an estimated genome size of about 1.2 GB. And we estimate it through k-mer analysis to have about 20% of repetitiveness. So we have this song sparrow population in the Mandarte Island in British Columbia. And this is part of a long-term study where we want to look at the island's population on inbreeding and inbreeding depression. So for this, we collected the social pedigrees from 1975. So you can see here at the right-hand side, they're being collected up to this date, actually. And then to this, since 93, we collected genetic data, so mainly macrosatellite data, which has corrected the social pedigree. So the aim really is to have a genome assembly so we can target SNPs that can help us elaborate on its genetic pedigree. So for this, we used mainly a short read assembly. So we use various Illumina reads types, so whether parent or mate pair, and to this NGS resulting scaffolds, then we added optical mapping. So we produced optical mapping for two different enzymes, TLE1 and BSSS1, and we had about a coverage of 500x, and we performed the de novo assembly for both enzymes. And these were the results. As expected, we obtained less scaffolds for TLE1 with a higher N50 number. And then from this on, we performed a two-enzyme hybrid scaffold, and the results of which you can see here depicted. So indeed, so we have an improved hybrid scaffold results whereby we have less scaffolds, so 118 in comparison to the 5,000 odd in our NGS scaffold. The N50 is increased, and perhaps the feature or peculiarity that I'd like to draw your attention is to this 44 scaffolds of the N90. So meaning that, and so if you think about, as a reminder, the karyotype that we're expecting of this bird is about 38 to 40 chromosomes. This probably indicates that we have some chromosomes assembled. And then secondly, we went on to and did the benchmarker universal single copy ortholog score, which is the Busca Scores. So the Busca actually attempts to provide a quantitative assessment of the completeness in terms of expected gene content of a genome assembly. And the results are really simplified into categories of either, you can see, complete genomes with a C, whether it's complete and single copy, complete and duplication, fragmented, and missing scores. So we had a very good Busca score, about 95%, which are complete single scores. And so we were very happy with his results. And so optical mapping did truly in this project increase the contiguity to up to chromosome level scaffolds and corrected 23 of the missed scaffolding assembly. And then on, we went further and looked at the linkage map to the song sparrow assembly. So we used linkage maps and used it against our song sparrow assembly, and we could identify very few incongruencies at this point. Furthermore, what we did though is looked at the homology between the zebra finch and the song sparrow, and we could actually see that in our hybrid scaffold song sparrow, we've almost got chromosome level as well, which indicated that these are extremely good results. And glad that Kaminish is at the moment working on these, and hopefully we'll get them published soon. So the second project is indeed again a short read assembly as well, but the main feature is that we used it not on a diploid species, but on a tetraploid species. And this was used with a finger millet, the Raji organism. This project is part of the Indo-Swiss collaboration in biotechnology, which prizes Professor Shimizu and Dr. Hatakayama from the University of Zurich, Professor Sriman from the GKVK Institute in India, and our institute. The importance of this plant is that it's a staple food in India, many countries in Africa, as well as Japan, and it's considered a superfood and highly nutritious. And a little bit, just to give you a little bit of a background on how we think this tetraploid species emerged, we believe that a few million years ago, we got the merging of this E. indica, which gave rise to the A genome, which is a diploid, and an unknown E genome diploid species. And then the species then went domestication into our cultivar. So the genome assembly workflow that we used was again a short read assembly. We used a very low coverage of long reads from Bio to get a higher N50 as well, and we produced optical maps using the DLE1 and the BSPQ, and we had about coverage of about 1000x. So for each of these labeled molecules, we then proceeded and produced a Genome assembly using, again, the DLE and the BSPQ1. Again, DLE gave us slightly much better results, or better genome assemblies than the BSPQ1, and performed a 2-enzyme hybrid scaffold, and then afterwards went and closed some gaps. And so these are results from the 2-enzyme hybrid scaffold from the finger millet. And what one can see here is, first of all, the number of scaffolds did reduce considerably, and we managed to increase the N50. Additionally, once we performed the Busca scores, we could see that we had about 97% that were complete. However, I'd like to draw your attention to this duplication level, which is about 60%. So it is believed that a polyploid species would have a duplication level of about 100%. And in the case of the finger millet, we only get 60%. So either we have misassemblies, and we're validating this right now using linkage maps, or the other, and we think the most plausible way is, what we believe is that most likely through evolution, because the species is a very old species, most likely genes or pseudogenes were lost through evolution, and that's why we only have this 60% on duplication level. Then we went on to the second part, which was the subgenome classification. So just as a reminder, we have a tetraploid species that originated from this merging of two diploid species. And therefore, what we wanted to do is really a subgenome classification. So what we did was we used SNP markers from short-read sequencing from the known parental species, which is E. indica. And then we sorted the diploid subgenomes based on the percentage of sequence identity, and then also the coverage estimation. So the high coverage was assigned mainly to the A subgenome, whereas the low coverage was assigned to the B subgenome. And then we went on, performed the hybrid scaffolds for this, and we could see that our N50 was pretty much consistent with the previous one that we obtained. So it is probably very likely that since we are dealing with these very distinct subgenomes, that these really did not interfere in the assembling process, and we could really separate them very nicely. And then we went on to look at the Busca scores. And here we could see that we have a complete about 90% for both A and B subgenome, and that the duplication level is actually extremely low, so 1.5%. And most of them are single copy hits. There are some missing hits in both A and B subgenomes, and we're working further on at the moment. So therefore, we did not only manage to assemble a tetraploid species, but also managed to pull the different subgenomes apart. And now what we're actually looking at is the environmental effect, so the impact actually of things like drought into each of the subgenomes and study stress responses. So for the third project, a bit of a tricky one because we started off with a short read assembly and then drifted on to a long read assembly, and I'll explain this in a minute. So for this project, we worked with Professor Bruno Studer and Dr. Dario Copetti from the molecular plant breeding group at ETH. And we worked on the Italian ryegrass, cultivar rabiosa, which is an important animal feed. And it's a fairly big genome, so it's about 2.5 GB organism, and it's a diploid species. To date, all of the available forage grass genome assemblies are either fragmented or rather incomplete. And we wanted to assemble this cultivar in particular. So we actually looked at the we obtained a short read assembly, and this short read assembly, when we looked at the specific regions, so for instance the difference between two alleles, we could observe that there was a lot of allelic variation whereby these alleles assemble really very separately. So if there were matches, you would assume to have all of them set in at this diagonal line. And you can see that there's a very low there are very low matches here. We have a high percentage of repeats and a lot of polymorphism between these two different alleles. So for this, then we assume that since we have such a huge sequence diversity between these two alleles, for instance, that clearly this pattern, we have to see this pattern in an optical map genome assembly. And that hopefully we will get both alleles represented in this optical map. So we therefore performed optical mapping using DLE1 only. And at the top, you can see the raw data specs. So we got very long molecules, about 300 odd kb in size, with a pretty high coverage. So here I just depicted that the organism, the haploid, is 2.5, but here we assumed that because of its sequence diversity, we assumed that it's a 5 GB genome, meaning that we would have about 85x coverage per allele. And when we did the de novo assembly here, we got about 260 maps, which had which are fairly big. And actually, the size matches the chromosomal arm size that we would expect for this species. And so we then went on. So from the molecular quality report and the de novo assembly report, we assumed that the data is quite good. But we wanted to further, before using the Bionano optical data, we want to further validate this to make sure that we could really trust the optical mapping. And for this, we did a validation using one of the things was the unimodal molecule coverage distribution. So we took 20,000 random sites and we looked at the coverage distribution, and I'll show you this in a minute, as well as looking at whether we had chimeric regions. And the way we looked at this was to manually inspect 4 regions with scores that were lower than 80 across multiple sites, and we found there that there was no indication of misassemblies. So from the total size of our de novo assembled optical map, which is about 4.2 and is matching the estimated diploid size, and also from the average coverage. So you can see from these 20,000 random sites that we plotted here, you can see that we have an average coverage of about 80x plus minus, so 70 to 80x, let's say. And we get this one single coverage peak. So suggesting really that our maps, so our Genome Assembly optical map, most likely is deployed, and we have few collapsed maps. So now that we trust our optical mapping, we then went on and looked at alignment to our deployed assembly. So, and here you can see that whether it's actually at a large scale, as you can see here, a large area of the genome, or at a small scale, we identify a lot of conflicts between the sequence and the optical map, so the NGS scaffolds and our optical maps. So basically, blue would be matches, and yellow would be just mismatches or conflicts. And so due to there's huge amounts of, so we had a lot of NGS conflicts, the optical mapping. So here, this, for us, it was important because Bionano optical maps worked very well in validating this, this assembly. And actually, it called a little bit for the importance. So we received this finished short-read genome assembly from a service provider, and we actually it really calls into that we need validating methodologies such as optical mapping, really make sure that the assembly that was performed is reliable. And so since then, so we have here, we did the hybrid scaffold, and you can see here that again we have a lot of yellow conflicts between the NGS and our optical map. Dario Copetti decided then to move on and assemble a long read and do a long read assembly. And here what we can see is there's a lot more of correlation between the NGS and the optical map. This is for exactly the same region of an optical map, the exact same optical map, and you can see that there's a lot less of misassemblies when using a long read assembly. And so this work is ongoing, and it's taken us, there was a huge delay just trying to figure out and trying to improve the short read assembly, and finally found out that there are far too many conflicts, and that is why we started from scratch long read assembly for this sample. So the next project I'll describe is a long read assembly that was done for the Apple cultivar Galaxy. So this project was done in collaboration with Professor Andrea Patocchi and Professor Bruno Studer, as well as the main lead of the project was Dr. Giovanni Brugini from Agroscope and the ETH. So this vegetatively propagated apple cultivar has, we expect, about 17 chromosomes and 700 MB, genome size of about 700 MB, and it's diploid genome. All of the available genome assemblies to date are homozygous genotypes, even if most of the apple genotypes are heterozygous. So and one of the features of apples is that mutations can accumulate in clonal lineages and even within branches of the same tree. So Galaxy actually originated from the cross of Golden Delicious and the Kit Orange and went subsequent bud mutations. And the aim of this project is really to produce a heterozygous diploid genome, and then afterwards use this diploid genome to study and look at structural variants in Gala mutants and cisgenic lines. And so for this, a little bit of a background. So we have indications from the previously published Golden Delicious genome that there are huge levels of heterozygosity, and that duplication can even span in that two alleles can be found on two different chromosomes. So we suspect a very similar scenario, and perhaps even a more complex so for our galaxy. So as I mentioned before, we performed a long read genome assembly, and mainly done with PacBio reads, and the reads, this quantic assembly was then polished using short reads. And in parallel, we performed optical mapping, again, at two enzymes, DLE1 and BSPQ1, labeled molecules, which then we performed the de novo assembly from each of them and gave these results. So especially for DLE1, we had a decrease in the number of scaffolds, and the M50 was pretty high. And we performed here the hybrid scaffolding, the two enzyme hybrid scaffolding, and these are the results that we got. So for the two enzyme hybrid scaffolding, so as a reminder, the expected genome size of Galaxy is 700 MB, indicating most likely that we have a deployed hybrid scaffold here in this case. We got low number of scaffolds, especially in comparison to the NGS scaffold. And as well, we got the N50 was also increased in our two enzyme hybrid scaffold for this sample. And to this, we added genetic maps, and these allowed us to actually anchor the scaffolds to chromosomes. And from this, we could actually obtain a primary haploid assembly, which was 719 MB in size, and a secondary haploid assembly, which is 600 MB in size, let's say. And so then we went on to look at the synteny between the primary and the second haploid assemblies that we obtained. And here on the y-axis is the secondary haploid assembly, the chromosomes, and on the x-axis we have a primary haploid assembly. And you can see here that there's a lot of heterozygosity in a lot of the chromosomes, indicating that these haploids are fairly different. And so this is at the moment the status of our assembly. So I'm happy to announce that this week it was submitted, this heterozygous Galaxy diploid genome, to bioRxiv, and hopefully will be published very soon. And you can observe here already that it's got a larger N50 than previously published genomes, Apple genomes, and as well the assembly size is also a lot bigger. And therefore, from this project, we really can say that we got diploid assembly. We could really manage to get the primary and the secondary haploids. Now, at the moment, what we're doing is adding phase information through graph-based approach using WhatsApp. And so, a little bit in the next couple of slides, I'll summarize some important aspects that we have experienced while using optical mapping for genome assembly. So one thing is indeed the nature of the genome, more specifically the allelic variation, do play a very important role. Because, for instance, in species or that are homozygous or homozygous-like, such as the song sparrow, so even if this is really a wild species, a wild animal, we suspect that there's a lot of inbreeding. And so if you think about having in a specific region of the genome, two alleles here represented, in this case, we would expect to obtain a collapsed optical map. And in cases where we have a lot of heterozygosity, so let's say the two alleles are extremely different, or just with very short spans of homozygosity, such as we encountered, whether it's actually at the subgenome level haploid, or just high levels of heterozygosity, as we observed in this species, we actually encountered that in most cases we obtain two different optical maps, one for allele 1 and the other one for the second. However, things start getting a lot more cumbersome when you've got long stretches of homozygosity, let's say, and these bubbles of heterozygosity that you find in the genome. So ideally, the assembling process should be disrupted when encountering these homozygous regions. And these would then give rise to an optical map upstream of this heterozygous region and another one downstream, which is a collapsed map, and then two maps for the heterozygous region, for instance. This would be the ideal case scenario, although you have a more fractioned, so the size of your scaffolds or of the maps would be a lot shorter. Also, in the case where you've got these bubbles of heterozygosity up and downstream of a homozygous stretch, you would expect that this homozygous stretch would then give rise to an optical map, let's say X, and that this is then spanned up and downstream by either optical map A coming from one allele and B from the other allele. And here then you've got again a more fractured optical mapping assembly, and maybe really the difficulty here relies in stitching these optical maps together, as you do not know what haploid. So optical map A is together with X, but then you do not know whether it goes C or D. The danger, because optical mapping relies on a non-haplotype aware genome assembly, then the danger is really that you end up with one optical map for these heterozygous regions, whereby you miss out some of the important heterozygosity that might be present in the sample. So this is a little bit items to think about when assembling your genome. Then, whether we use a 1 or a 2 enzyme hybrid scaffold, we have observed that here are we looked at the comparisons between the 1 or 2 enzyme hybrid scaffold. So it's clear that the choice of the enzyme that one uses will be really determined by the underlying genome that you're working with. So first of all, you have to have an appropriate number of labels per 100 kb, and this should be within the specs of Bionano. So in general, the DLE hybrid scaffold alone did yield very good and contiguous and high quality de novo assemblies. But we found out that there was an increasing number of projects that decided to go on into using further complement this and use another enzyme. And there has been, as in this example, so one could see that we've got less unscaffolded NGS in our final hybrid scaffold, or it can be because the N50 rises a bit more as well. And I've not shown the results for the apple as well, but we also observed about the N50 to increase 25% when adding the second enzyme, and about also the largest scaffold also increased about 11%. We also encountered cases where we added a second enzyme just because the DLE was not covering specific chromosomes in their species. For instance, they had very few labels per 100 kb in certain chromosomes. And for this purpose, we had to target these chromosomes using a second enzyme or a second nicking enzyme. And so a little bit to conclude here. So we have used Bionano optical mapping for a wide variety of species that range in different sizes. And one key point really for a successful hybrid scaffold if you want to use Bionano optical mapping, is really to have a high quality NGS draft, which then reduces the conflicts that one finds between the NGS assembly and the optical maps. For sure, long read assemblies provide very good results, but we've also encountered very good results using short read assembly strategies as well. We could use Bionano actually as an orthogonal validation strategy, as in the Rabiosa species, to just validate a genome that was provided by a service provider and was bought as a package. And we could use Bionano as well, optical map, to assemble polyploid genomes, as in the case of the finger millet. And we have the feeling that the two enzyme hybrid scaffold may give you some more power to integrate some more of the NGS scaffolds and also target regions that DLS is missing, perhaps. So, in general, I would really say that the Bionano optical mapping really improves our genome assembly greatly. And one has to really, though, take into consideration the complexity of the genome. So you cannot forget that we have a ploidy here, or the repetitiveness, how much of it and how big is it, and also the heterozygosity and the degree of a heterozygosity, especially because we're dealing with a non-haplotype aware assembly with the optical mapping. And so with this, I'd like to thank my boss, Professor Ralf Schlappbach, and my colleagues at the Functional Genomics Center for their input and help, as well as the Bionano Genomics team for their incredible support as well, and the university and ETH scientific community, as well as our collaborators, which from the project I have just presented, especially since a lot of this data is still unpublished. And with this, I'd like to thank you for your attention, and I'm happy to take any questions.
Thank you, Lucy. That was indeed a great presentation. Thank you for sharing your experience with us. I have a first question for you. You have been working with several sample type here. What is the key for a successful Bionano assembly? Do you have some insight on that?
The key really is, so in regards to the hybrid scaffold, is really to get a high quality NGS draft. And this can be obtained by getting really high quality data, really enough coverage. Also, one has to think about that you need enough length in order to deal with the repetitive regions. And also, if you think about a long read assembly, we need to do some correction prior to the assembly, and before doing a hybrid scaffold, for instance. And that's why we used short reads for polishing. And also, the parameters that one uses in the different assembly tools have to be appropriate. So indeed, for a hybrid scaffold using Bionano, we really need this really high quality NGS draft to start with in order to make the most out of this technology.
I see, thank you. And during manual auditing or annotation, how do you differentiate allele heterozygosity with assembly errors? What would you recommend?
Okay, so that's a really good question. So allele heterozygosity, so let's say it is usually quantified using alignments against reference gene sets, like single copy marker genes, for instance, and you can also do coverage analysis of the aligned regions. And then the misassembled regions usually can be identified by incorrect pairing of, for instance, of Illumina reads or coverage drops as well. And one can also think about, so isolated heterozygosity normally is more likely to be an error, really. And if it falls in a coding region, then the codon still needs at least to make sense. And if there's a disruption of what would be a heavy protein on a normal sample, then I would then suspect that this most likely would be an error, actually. Thanks.
Yeah, I think this is most useful, Lucy. Thank you. And if you could choose, what is a new feature or function in Bionano system that you think will benefit genome finishing community?
So of course I've got a huge wish list, but I think the most important one, and the one that most people would benefit from, would be a haplotype-aware genome assembly. And this would be extremely desirable because nowadays there's a lot of attempts to phase our genome assemblies. And what we find is, and this is one of the examples where we're dealing with cassava, or one of my colleagues, if you phase an NGS assembly and then you add optical mapping, we find more conflicts than if we use a non-phased NGS assembly and add the optical mapping, just simply because optical mapping is not phased. So a haplotype-aware genome assembly would really be definitely an asset and really be extremely helpful. Additionally, of course, other DLE enzymes. So as you can see, we're using a combination of enzymes to do the hybrid scaffolding. So a DLE enzyme, another one with another pattern would be extremely useful. And if it's labeled with red, it would even be better then, because we can pull, theoretically, the sample that was prepared by two different enzymes with two different colors into one compartment or flow cell of the sapphire chip, and make really use of this 1.3 TB of data that we could get nowadays and run it in parallel. And so additionally, we've got a huge wish list, but also perhaps an SP protocol for plants as well. That would be extremely useful. So I think Bionano, it's helping a lot the plant community. And yes, I think an easier protocol to extract high molecular weight DNA would be definitely very useful as well. Yes, but thank you.
Yeah, that's great. We will keep in mind your wish list, and we are already work to address some of those points. So thank you again, Lucy. I wish we would have more time to discuss all these topics. Again, it was great to have you here today to discuss your genome finishing work with us, with our Bionano platform. And thank you everyone for tuning into this webinar. Please email any question to support@bionanogenomics.com and reference this webinar. Also, please check our website at www.bionanogenomics.com for more information and resources. And bye everyone. Have a great rest of the day.
Thank you very much.